Hi. So, in our last discussion, we did a bunch of numerical simulations to understand a few features of genetic drift, and I promised you that in the next one, which is this one, we are going to look at some examples and a few features. So, before we get into that, if you remember, I had made a statement which was that in the context of drift, the probability of fixation of an allele due to drift alone is equal to its starting frequency. And we had done some simulations and we had seen that this was looking in general to be a correct statement. So, today before we get into the examples, I will give you the demonstration of why this property is true. It is a very simple thing, it is just a bunch of logical statements connected to each other. So, assume that we have a diploid population which has got n individuals, and in this particular population let us assume that drift is the only evolutionary mechanism operating. So, there is no mutation, there is no migration, there is no selection. Now, since the diploid population has a size of n, the total number of alleles in the population has to be equal to 2n, right. Now, let us make an assumption that each one of these 2n alleles are different from each other. So, each one is an unique allele. So, if that be the case, what will be the frequency of each allele? Obviously, the frequency of any given allele is going to be 1 by 2n. Now, when we say that drift is operating, by definition drift is unbiased, right. So, since drift is unbiased, therefore, all these 2n alleles, each one of them have an equal probability of getting fixed. If that be the case, what is that probability going to be equal to? That probability is going to be simply 1 by 2n, right. Now, let us assume that all these alleles are not different from each other. Let us assume that there are two kinds of alleles, A1 and A2, such that there are x copies of A1 and say y copies of A2, right. So, x plus y is equal to 2n. Now, if there are x copies of A1, then what is the overall probability that a copy of A1 is going to get fixed? That is simply going to be x into 1 by 2n, right. Now, if that is x into 1 by 2n, then also note that x into 1 by 2n is also the starting frequency of A1, right. Therefore, we have proven that the probability of fixation of an allele due to drift alone and nothing else is going to be equal to its starting frequency. It is a simple logical thing, nothing more than that. Okay. So, now we get on with our examples. Now, when it comes to drift, there was a very important, very famous experiment done in 1956 by this person called Bury, and this is a absolute textbook example. Every single textbook of evolutionary biology has a very high probability of having this experiment. So, we will spend a little bit of time in understanding this one. So, this is an experiment using Drosophila melanogaster, and what Bury did was he founded 107 Drosophila populations. Each one of them had 8 pairs, which means 8 males and 8 females. Then these populations were not founded in random. They had a property which is that every single founder, so 107 into 16 flies, all of them were heterozygous at a locus called the Brown locus or the BW locus. Now, at this locus, the BW gene, it codes for an eye color gene, and therefore, the beauty of the experiment is that all the 3 genotypes are discernible only by looking at the phenotype. So, you look at the eye color and you know that if it is a white eye color, then it is a BW homozygous. If it is a bright red orange eye color, then it is a BW75 homozygote, and if it is a light orange color, then it is a BW75 heterozygote. So, what he does is he starts the entire experiment with the heterozygote. So, everybody is light orange in color in terms of their eye color. Now, the very important reason for choosing this particular gene and nothing else is that there is no known fitness effect of these 2 alleles. Why is this important? Because remember, we are studying drift alone and therefore, we do not want selection to mess things up. and all he did in the experiment was that every single generation he counted the number of genotypes in the population, and every single generation he again collected 8 pairs, 8 males and 8 females at random, and then used them to found the next generation. So, basically the eggs laid by these pairs, those are the ones that created the next generation, did this for 19 generations. So, here is what the experimental data looks like. Now, I will tell you how exactly this graph is drawn. So, each panel that you see is for a generation. So, you can see it said initial population, generation 1, generation 5, etc., all the way up to generation 19. Now, what you have on the x axis is the number of BW75 alleles, and what you have on y axis is how many populations had that particular number of BW75 alleles. So, it is a kind of a histogram. Now, remember, every generation the population size is being kept constant at 16, 8 males, 8 females. So, since there are 16 individuals, then for the 16 individuals you are going to get 32 alleles, that is all you can have. Therefore, the number of BW75 alleles can vary from 0 to 32, that is the range, and every generation he is simply counting how many populations have 0 BW75, how many have 1, how many have 2, how many have 3, all the way up to 32. So, I will walk you through this panel slowly. So, let us look at the first one, okay, the first panel over here. Now, if you look at this panel, you see that generation 0, all the populations have 16 BW75 alleles. Why? Because all of them are heterozygote, right. So, all 107 have 16 BW75. What happens in generation 1? Drift has started operating because the population size is very low, and now the populations have different numbers of BW75 alleles, and you can straight away see in the second panel that the numbers have deviated from the value of 16. There are some which have about 14, there are some which have about 22 or 24, and so on and so forth. You go to generation 5, you go to generation 10, now you can see that the number of BW75 alleles in the population is varying a lot, which is why you are seeing the histograms getting spread out, and generation 10 is when the populations start getting fixed for either the BW allele or the BW75 allele. How do I know that? Because generation 10 is when for the first time we are getting some populations which have 0 or some populations which have 32, and then as time progresses, generation 15 and generation 19, you can see that the bars on the two sides, they are becoming taller and taller, and the ones in the middle between these two extremes, they are becoming smaller and smaller, right. Why is that happening? Because as time progresses, more and more populations are losing the BW75 allele or getting fixed for it. So, by generation 19, as you can see, 30 populations completely lost the BW75 allele, whereas 28 populations were completely fixed, which means that every individual had only the BW75 allele and nothing else. 28 were of that type. So, the reason that this particular experiment is so famous is because very nicely it shows you the two major properties of drift that we saw from numerical simulations. One is that due to drift alone, if you start with replicate populations, then all of which have the same genetic makeup, due to drift alone over time these populations can diverge from each other. So, that is exactly what you are seeing, that you are starting with just one tall building and how that thing is now getting distributed over time across various number of BW75 alleles. And the second major thing it shows is that the ultimate fate of drift is either fixation or loss, which is what is happening over here. By generation 19, you can see that 58 out of the initial 107 populations, which is about 50%, slightly more than 50%, that has become fixed in one direction or the other. So, we will get back to this particular experiment somewhere in our next discussion, where we are going to when we are going to look at how to measure drift, we will see what is the measurement of drift in this particular case. However, before we get there about measurement of drift, we need to talk about two special cases of genetic drift, and these two special cases, the first one is called founder effect. What is founder effect? So, suppose you have a population which has some genetic composition, some allele frequencies, and then from this population a small subset comes out and goes and settles somewhere else. And because it is a small subset, there is always a probability that the genetic the allelic frequencies in this subset is very different from the allelic frequency in the main population. And now, when this small subset goes and settles in a new place, and then over there the population size expands, then after some time that population is going to have a very different genetic composition compared to the ancestral population. But that need not necessarily have anything to do with selection or any stuff. That might simply be because the founders who began that new population, they just due to sampling error had a very different allelic composition to begin with. So, just to give you a example, pictorial example, suppose you have a situation like this, where you have, let us say, blue circles and red squares in some frequency, and let us say we had 3 different founding events from this. In one case, just due to sampling alone, you only have red squares founding that population. In another case, again due to sampling alone, you have blue circles founding the population, and in the third case you have again some red and some blue. Now, after some time, if you are not aware of the fact that the founding happened this way, you are going to look at these populations and say, aha, look at this top one, it is fixed for red. That means that the red allele is giving some major selective advantage over here, because of which this population is fixed for red, or similarly it is fixed for blue, and same logic. However, the real reason for which this has happened is something much simpler, that is because when they started off, they started with a different frequency, all reds or all blues, and that is why they are all red and all blue today. The second very interesting case is actually just a variant of this, which is known as the bottleneck effect. So, in the case of founder effect, you have one population being the ancestral population, and from that a small subset going somewhere else. In bottleneck effect, nobody goes anywhere, you have the large population, ancestral population, and due to some reason, some environmental catastrophe or some political stuff, whatever, the ancestral population's size becomes very small. And when the size becomes very small, then it is entirely possible that the allelic frequency changes again due to sampling error and nothing else. So, just to give you an example, so suppose you think about the ancestral population as a bottle full of 3 different colors of marbles, green ones, orange ones, and deep orange ones and light orange ones. And let us assume that the population size has shrunk, which is represented by the narrow neck of the bottle. And now you shake the bottle a bit, some marbles come out, some beads come out, and that represents the fact that a small subset is what is left due to your whatever environmental catastrophe or whatever, and the surviving population now has a very different composition, which is what you see over here. The bright orange marbles or the beads, they have been completely lost, and now the population only consists of green and orange ones, light orange ones. So, again, if you look at it and you are not aware of the fact that this kind of a constriction has happened, bottleneck has happened, you are going to think, aha, the bright orange allele had some major fitness related issues, it reduced the survivorship or fecundity in a big way, and therefore it has been thrown out of the population. But in reality, that is not what really has, that is not what has happened. All it has happened is again a kind of a sampling error. Now, there are a few points that I need to make in the context of founder effect and bottleneck effect. I have been sitting in interviews at IISER Pune for last 17-18 years, and one of the common questions that we ask to people is what is genetic drift? And many students end up saying, oh, genetic drift means founder effect and bottleneck effect. That is not really true. As I am showing you, founder effect and bottleneck effect are two special cases of genetic drift, sampling errors happening in two particular ways. But these are not the only two ways in which genetic drift can happen. As we saw in our simulation, genetic drift can happen even with constant population sizes as long as that population size is finite, and therefore, genetic drift needs to be defined as those random change in allele frequency and not as founder effect or bottleneck effect. This is a very important message that I need to give to all students. Appreciate the correct definition of drift. The second point is that, as I said, under the hood all that is happening is a sampling error, and the sampling error is happening in two cases because you have a large population. From that large population you suddenly have a very small subset, and that subset may not be representative in terms of its allele frequencies of the main population. Therefore, although people tend to differentiate between them, in terms of their genetic signatures they are often very same, and until and unless you have special information, it becomes very difficult to tease apart whether a particular signature that you are seeing is because of founder effect or a bottleneck effect. And finally, the main stuff that happens over here is that the population genetic composition of the offspring generation is very different from the parental generation. So, the because of the sudden reduction in population size, and the main thing that ends up happening is typically there is a reduction in the genetic variation. Okay, that is the main thing. Founder effect and bottleneck effect, both of them typically lead to a reduction in the genetic variation. I mean, so does genetic drift, but founder effect and bottleneck effect are very stark and very one step big ticket effects that you can see. So, let us look at a few examples of both founder and bottleneck effects. So, this is one of the very famous, again big examples of founder effects. So, we know that in humans, Homo sapiens, we originated somewhere in East Africa, and then from East Africa we actually migrated to different places, and these migrations did not happen in one event, they happened subsequently. So, we went from point A to point B, and then after some time from point B one group went to point C, another group went to point D, and so on and so forth. So, as far as scientists have been able to trace out, so human beings originated, you can see the yellow star here, that is Addis Ababa somewhere in East Africa, and then from there were lots of migrations events that happened towards other parts of Africa. These are what are represented as these red inverted triangles. Then there was one migration event that happened towards Middle East, going towards north of Europe, Russia and all that, and then from the Middle Eastern population, one set went towards Europe, Western Europe, another set went towards Asia, East Asia, and then from there, from the China and other places, there were other migrations that happened, went towards northeast of Russia, eastern China, Japan, etc. And very importantly, this the Middle East group, this sent out one branch towards Far East Asia, and from there they went towards Indonesia, Philippines, all those places, and from there they went into North America, and from North America they went into South America. So, this is how we have been able to piece together the migration routes of ancient human beings over time. Now, appreciate one thing, that each time a group is going from one place to another, you are going to get a founder event. So, for example, when they go from Africa to Middle East, that is one founder event. When they go from Middle East to, let us say, Western Europe, that is another founder event, and so on and so forth. So, if that be the case, what we have is a series of founder events. And what I told you is that each time a founder event or a bottleneck event happens, typically the genetic variation goes down, right. So, what people did was these people, Liu et al., they ended up looking at the genetic variation in people from these various parts of the world, and they ended up showing that as you go further away from East Africa in terms of the migration, the genetic variation that is present in the humans, it keeps on going down. So, the maximum genetic variation actually is found amongst the human beings in Africa. Then the second largest you find in amongst people who are in the Middle East. From there, the amount for people who came to India and other parts, it goes down. And then when you look at people who went to Far East Asia, these are the red squares on this, it goes down further. And then when you look at those individuals which went into Indonesia, Philippines, etc., their genetic diversity goes down further. And then if you look at the individuals, what is shown on this map in white circles, these are the guys who are in North America and South America. These are the so called last in terms of the founder events. If you look at their mean heterozygosity, and I will tell you what mean heterozygosity is, it is a measure of how much variation that is there in the population. If you look at that, you see that it decreases further. So, in other words, you see a very nice negative correlation between how far you are from the center of origin of humanity and what is the level of genetic diversity that you have. Okay. So, this is a very nice example of what continuous founder events can do. Now, one can of course ask that this might have been due to selection. How do we know that this is not due to selection, this is due to drift alone? That is because the particular genetic sequences where they are checking out the variation, as far as we know, these are neutral sequences. In other words, sequences which do not have any effect on the fitness and therefore is not seen by selection. Hence, we know that this is due to drift alone. Okay. This is big ticket, entire humanity taken together. Let us look at a more specific example of founder effect in humans. Now, to people in India, this gentleman I am sure needs no introduction, Bollywood actor Hrithik Roshan. Now, many of you might be aware that Hrithik Roshan has a condition which is known as polydactyly. He has an extra finger over here, right. Now, turns out that polydactyly is actually pretty rare amongst humans, and its normal frequency is about 4 to 12 per 10,000 births. However, there exists a population in the US where the frequency of this particular condition, polydactyly, is much greater. And how much greater? This is about 7 per 100 births. So, this population is the so called Amish population. So, the Amish are a known migrant community. So, they migrated from Switzerland in particular to the US somewhere in the early 1700s. So, these are a fascinating people because they believe in staying away from modernity. So, in many ways these people are retaining many of their cultural practices from 300 years back. They typically do not use any machines, most of they believe in very hard work, extremely God fearing people, and most importantly they do not marry outside. So, the Amishes marry other Amishes, and because of all this, the genetics of the Amish population is absolutely fantastic, and there are lots and lots of studies on the genetic properties of these people. But as I said, the one thing that is very interesting among the Amish is that they have a very high frequency of polydactyly, and this particular thing actually comes due to a single mutation on a gene known as the EVC gene, right. Now, why exactly is it that the Amish have such a high frequency of polydactyly? So, one of the things that the Amish do is that as part of their culture they maintain very detailed genealogies. Therefore, we essentially know who is the father of whom and who is the grandfather of, and who is the great grandfather, etc., etc. So, scientists have actually traced the entire genealogy of these Amish migrants, and they have figured out that in 1744 there was one couple, one Mr. and Mrs. Samuel King, who came from Germany to Pennsylvania, and it is not clear whether Mr. King had it or Mrs. King had it, but it is pretty clear that one of them had this particular mutation, it is a recessive mutation, and after that this one mutation essentially ended up spreading amongst the Pennsylvanian Amishes, and the entire problem that we have today, 7 per 100 births, is because of that one couple. In other words, if those two people, the husband and wife, if they had missed their boats, or if the boat had sunk, God forbid, during the transatlantic cruise, then the Amish people would not have had this particular condition. Now, I am talking in terms of polydactyly, but actually what they have is a bigger condition than that. It is a condition called EVC, and because of that there are many other physiological problems that the Amish are saddled with, and as I said, all because of Mr. and Mrs. King coming in 1744, a massive example of, and a very well known example of, founder effect in humans. So, now we go to bottleneck effects, and for bottleneck effects we are going to go to Micronesia. Now, I am sure many of you would not have heard about Micronesia. So, the Micronesia is it is a bunch of islands, as you can see, somewhere north of Philippines, and very small tiny islands, and amongst these islands that constitute this nation of Micronesia, there is one which is known as Pingalap. Okay, and I am showing you a satellite picture of Pingalap just to tell you how small it is. Okay, you are not getting an idea of how small it is, but you can see that somewhere here, somewhere close to the center, you can see a strip, right, a white strip over here. This is a runway. Okay, just for scale, this is a runway. So, this tells you how small this entire place is, right. Now, why is this place interesting? Why is this place so well known in whole of biology? That is because there is a certain condition known as achromatopsia. What is it? It is a eye disease which leads to complete color blindness. Now, what do I mean by that? See, normally when you have color blindness is about inability to distinguish between certain pairs of colors. So, let us say red green color blindness or blue yellow color blindness, people cannot distinguish between that pair. In the context of achromatopsia, they have complete color blindness, which means that they cannot distinguish any color, they see the world in black and white, okay, that bad. And more importantly, if it were just seeing in black and white, it would have been bad, but in reality it is way worse than that. These people, their visual acuity, which means that how sharply they see something, is really bad, and more importantly, they cannot go out in sunlight. Okay, the moment they go out in bright sunlight, and these are tropical places, so obviously the sun is really bright over there. The moment they go out in sunlight, their eyes start watering and paining like anything, right. So, obviously this is something, and if you are in an island, okay, this is not like a highly developed kind of a society where you have lots of ways to not go into the sun, etc. These are economies which depend on nature, and therefore not going out into the nature during the entire daytime is a big problem for these people. So, now obviously the question is, how did it come to be? So, turns out that achromatopsia is known outside Pingalap as well. However, if you look at outside, the prevalence is very small, 1 in 20,000 or 1 in 50,000 is the worldwide rate. However, if you look at the Pingelap island, for those guys 6 to 10% of the population have this condition and about 30% are carriers. Now, how come something which is so problematic for the species, how come it is so widespread? And turns out the story over here is fantastic, or not for them, but from a scientific point of view. So, turns out that in 1775 there was a very massive typhoon called Lienkieki. I hope I am pronouncing it right, Lienkieki, right. Now, due to this typhoon, about 90% of the population of that entire island was wiped out, they just died. And after the typhoon was over, all the vegetation and everything was affected. So, there was basically no food for a very long time, there was almost a famine like condition, and a huge number of people died due to starvation after that. So, finally after this entire disaster got over, there were only about 20 people left on that island, and the current population of Pingelap, which is about 250, they are all descendants of these survivors. Okay, 250 is the population now because lots of people have migrated outside Pingelap, but the population had actually gone up much higher. I think 750 to 800 at some point it had gone up, and even every one of those were actually descendants of these survivors. Now, turns out that one of the person who survived was the local ruler, and also turns out that this person happened to be a carrier for a recessive mutation for a gene known as the CNGB3 gene, and this particular gene encodes one component of a protein which is crucial to the functioning of the cone cells in our eyes. So, you know that we have rod cells and we have cone cells, and you know that cone cells in our eyes are the ones which are important for color vision. So, the reason they are important for color vision is because of proteins, and one of the components of that protein, the gene coding for that, gets a mutation. That is it. Because this person survived, and because he was the ruler, probably he ended up having more number of babies, relatively speaking, the condition spread, and today the Pingelap population is suffering from this kind of a situation. So, just for fun, I want you to watch this particular short video. It is just 2-3 minutes video. It takes you to the Pingelap atoll. It is beautiful, absolutely. I mean, natural beauty is superb, but it focuses on the condition and how painful it is for the people of that particular place. Note that I did spot a few errors in the facts presented in the video. So, if you compare the facts over the stated in the video with what I am telling you over here, you will see some discrepancies. I think mine are correct because I ended up cross checking my facts with multiple papers. I do not know whether they have done any of that. The second thing that I want you to look at is this very famous book. In fact, it is this book which made the Pingelap condition famous all over the world. This is the book called The Island of the Colorblind by Oliver Sacks. Okay, brilliant author, has written on multiple topics related to evolution and many other things. This particular book I have read excerpts of it, absolutely brilliantly written, highly recommended. And finally, if you want to learn about genetic drift, then the Amoeba Sisters version of genetic drift, this particular video is really good. The information content is roughly similar to what we have discussed till now, but obviously the way they present it is very fun. So, till this point we were just talking about drift changing the composition, but at some point we have to be able to measure the effects of drift, because without measuring we can't really figure out what exactly is going to do to evolution. And how to measure drift and what will that measures lead to, those are the things that we are going to do in the next discussion. See you. Bye.